The purpose of this set of bank of slides is to introduce you to the concepts of health effects in environmental health. So let's get started. Now, as you can see that at any given point in our life, at any given point in our health states that we are experiencing, that is predicated by two factors. The first factor is genetic component, which means the genetic makeup of every individual, the genes that we inherit from our parents, and the genetic products that are present in our body at any given point in time, and their activities. So this, for example, could be receptors in the various tissues on our body, and the various gene products, for example, levels of proteins that are present. And on the other hand, the at any given point in time, the health state determined or depends on the environment that we live in. So if it is a particularly toxic environment, then the toxicity of that environment will overcome any kind of genetic resistance that we may have developed. So, for example, under extreme conditions of air pollution, for example, we feel sick. And whether or not we could have been otherwise protected because our genetic endowment doesn't matter anymore. So which means that at any given point in time, we have both a genetic endowment and an environmental component that determine our state of the health or the mind. Remember here, health, of course, is used in the same sense that was stated by the World Health Organization's original definition, that health is the complete a state of complete physical, mental, and social well-being, not merely the absence of infirmity or diseases. So from that perspective, you can see that both genes and environments are very important in determining our health state at every point in our life. And having said that, therefore, diseases, illnesses, health states can evolve both over time and can be found over a spectrum of conditions. So, in other words, if we were to construct some sort of a conceptual diagram, you could see that on one hand, we could say that a particular illness could be very acute or not very acute. The word acute here basically means that whether they have developed over a short period of time, that is, whether it was whether there was something that was that just developed recently or that took a long time to develop. So that's acuteness. That's how acuteness is defined. On the other hand, you could also have another axis that could tell you that whether it's very severe. For example, if a health condition is such that you feel extremely unwell, you feel unwell and there are some major destruction of the various organ systems of the body, then that's not a very good news. So severity is defined from that perspective, the extent of damage that the body has sustained. So therefore, if we put these two concepts together, we can find or we can construct some sort of a quadrant, if you will. In one quadrant, you can see that there are some diseases that are chronic and that are less severe. For example, chronic depression. Chronic depression takes a long time to develop, and that may or may not be very severe. Another example might be very low-grade exposure to inorganic arsenic in drinking water, for example. When that happens over a long period of time, and when I mean not long period, I mean basically over say decades of exposure, but extremely low concentrations of inorganic arsenic, then there may be skin pigmentation, but they're not very debilitating. Almost all of us are exposed to some environmental contaminant over our lifetime, which is a chronic and over a long period of time. Many of us do not manifest any in any disease conditions, although we are exposed to environmental conditions, various environmental toxicants over a longer period of time. So this belongs to a chronic, less severe kind of a spectrum, end of the spectrum. At the other end of the spectrum are acute and severe disease. For example, consider those situations where a lot of people, almost 3,500 people, died instantly in Bhopal in 1984 when they were exposed to very high concentrations of methyl isocyanate gas that leaked out of the pesticide plant, the Union Carbide pesticide plant in Bhopal. Now that is a very classic and probably a dangerous example of an acute and severe health condition taking place as a result of an environmental exposure. More commonly, however, you can have an acute condition but which is less severe, or chronic conditions but that are quite severe. For example, an example of an acute and less severe disease conditions or disease or health effect would be influenza. Many of us fall sick within days of contacting a person or coming in contact with a person with influenza. This makes us sick, and we may remain bedridden for a few days with symptoms and signs of flu with high fever, cough, respiratory discomfort, etc. They develop very rapidly, therefore they're acute. But they may or may not be very severe. They may or may not be life-threatening. They can be mild discomfort to some extent. On the other hand, you can have a chronic and severe conditions. The chronic and severe conditions are somewhat rare, but they occur under circumstances where a person may have developed this over a period of time, but all of a sudden it becomes very severe. For example, somebody has been exposed to a carcinogen for many years, and the carcinogen has not manifested in the form of any disease, but when it manifests, sometimes it manifests in the form of very, very late stage of cancer. At that point, one cannot do much, and the disease manifests in the form of high severity, grave severity, and a person can die within a short period of time. So you can see that if you think in terms of the acuteness of the emergence of the disease and the severity of the disease, there can be almost like a spectrum that we must consider. However, it is also true that diseases, particularly following exposure to environment, can take a fair bit of time to develop, and there were a couple of stages. And here are those stages that we are going to learn a little bit. When the exposure begins, as you can see in the leftmost side of the figure, disease does not instantly manifest amongst us or in us. It takes a while before that environmental exposure can come to a point where disease actually begins biologically within the biological systems. And the interval of time that lies between an initial exposure to the exposure agent of interest and emergence or beginning of the biological causation of the disease is known as induction period. This induction period can vary a lot depending on the particular environmental exposure agent under study. For example, when people are exposed to relatively high concentrations of inorganic arsenic in their drinking water of the order of 50 micrograms per liter or more, then these people are prone to develop different kinds of diseases, ranging from skin lesions to skin cancers to cancers of the urinary bladder to lung cancer and other conditions. However, the period of time that elapses between an initial exposure to inorganic arsenic and the initial origin of the disease is virtually unknown, but it can be presumed that it is a long time, because cancer, for example, always takes a very, very long time for them to develop. Therefore, you can argue that the induction period of an environmental carcinogen is fairly long. And then again, between the initiation of the process of the exposure variable in the environment causing a biological damage that will eventually result in emergence of the signs and symptoms of a disease can be fairly long, which is known as a latent period. Once again, the example of cancer is a good example to say that between involvement of a single cell, etc., that becomes carcinogenic, and then it starts spreading into the other cells, and emergence of a damage to the body, because the cell then starts growing and grows uncontrollably and replaces the stroma of wherever it is in the body, and then the manifestation of the disease starts. It may take fair bit of time. And people have tried to work out what that length could be. Sometimes it is 3 years, 4 years, 5 years, etc. So that period is known as latent period. And obviously, latent period and induction periods can vary depending on the environmental agent that we're talking about. For example, if an environmental agent is air pollution, and if the health effect that we are interested to study is, for example, cough and cold and cough and irritation, etc., or irritation in the eye, then if it is a very severe case of air pollution, then the induction period and latent period is very, very short. On the other hand, if we are interested in studying environmental mutagens and cancer, then you can see that the latent period and the induction period can be very, very long, because cancers typically take a long time to develop. So for example, if we are interested to study exposure to inorganic arsenic and emergence of cancer, for example, it can take a very, very long time, both in terms of induction as well as latent period. So therefore, because we have seen that both genes and environment are important, and there is a role of therefore genetics, genetic endowment, susceptibility to environmental variables. Let's talk a little bit about what could be the role of genetics in environmental diseases. Now, there are several different ways in which genes can be tampered with or interfered with, or genetic functions can be interfered with. Genes, as you know, are like strings of 3 nucleotides, and nucleotides are kind of nitrogenous bases, and these bases are named as adenine, guanine, thymine, and cytosine. These are present in the DNA of our that is in us, and the DNA is packaged in the 23 pairs of chromosomes that we all carry. And genes can be interfered with in a number of different ways. Probably the simplest way and the most common way in which genetic functions are altered is by altering the structure of the specific genes. And at the simplest, it could be just a substitution or addition or alteration of the structure of very simple single nucleotides. When this occurs in more than 1% of the cases, or 1% in population frequencies, then the specified term is single nucleotide polymorphisms. Sometimes, however, genes are present, genetic changes take place, and genetic changes, the structural changes in the gene lead to functional changes in the proteins that they code in terms of the different kinds of enzymes and receptors that they are associated with. But they occur rarely, for example, less than 1% of the cases, and this is known as mutation. Now, environment and specific environmental exposure can cause both mutations as well as single nucleotide polymorphism. And for almost every disease that are known to human beings, there are some contributions of an environmental agent that interferes with the genes in either causing single nucleotide polymorphisms or mutations that then lead to different kinds of gene products, in turn leading to manifestation of the disease. This is a very hot area of research, and you will almost on a regular basis read about research reports where single nucleotide polymorphisms of one or the other disease have been identified or mutations have been studied. Environmental mutagenesis is a specialized field of study where the object of the study is to study what are some of the environmental variables that can change the genetic constitution of the body, leading to specific disease conditions. So from here on, let's move on to a little bit technical issues as to how do we measure health effects, both in individuals and in populations. For individual level measurements, which is usually conducted by health providers such as doctors, nurses, health workers, and other professionals, there are some specific ways of doing these things. So the focus here is on individual symptoms and signs. Symptoms are referred to as those situations where people express that they're unwell. So for example, if I say that I've got headache, or if I've got chest pain, then that would be termed as a symptom. Signs, on the other hand, refer to the situation where a health provider, using tools of measurement, will find out that there are certain specific manifestations of a disease that is present in the body. For example, appearance of a rash, or increased temperature, or high blood pressure. these are all signs of diseases. So collecting individual symptoms and signs through history taking and physical examinations are some of the ways in which doctors, nurses, health providers, health professionals will find out about the health effects that an individual is suffering from. On the other hand, when we talk in terms of health effects that can be gleaned from population-based data, they're usually done using data analysis from surveys, from publicly available data, from registries, and from linked data. So take a look at the linked data for a while. A linked data is actually a situation where two or more databases are linked with each other or connected with each other by considering an index, a series of index cases or a tuple of index cases. For example, in most such situations, at least one data source contains information about exposure variables, and one or more data sets will contain information about the health outcomes. A very common application that you can see quite frequently in the literature is how people relate air quality and health issues. In studies that relate air quality in a particular city and health conditions, perhaps admissions due to, say, asthma or heart disease, are usually carried out by linking two data sets. One such data set would be meteorological data of air quality collected from the meteorological stations or weather stations or by coring the meteorologists. And the health data can be obtained from hospitals of individual areas by visiting the hospital records and identifying the number of admissions because of specific health conditions. And then these two data sets are combined together based on the geographical location of the specific hospital and the specific weather station, where these two things have got a common unit of geographical location. Then those two data sets can be combined together, and then you can do further number crunching and data analysis. Now, of course, there are other sources of morbidity and mortality data in public domain and for public health purposes that can be used as well. We have already mentioned about the importance of hospital records in particularly understanding association between environmental variables and specific health conditions. One can also use death certificates that are issued by the health systems to go and study the rates of death over a particular period of time. Employment and school records are very, very important sources of data for conducting prospective or retrospective cohort studies, because many people, when they first get employed or first seek employment, their companies or their offices will conduct a health checkup, and in those health assessments, a number of different parameters are evaluated. So they're very good they're very good sources of information. School records are also good sources of information of health status data. At the least, they provide information about the absences that a pupil had, or specific illnesses that a student complained of, or maybe and so on. In addition to a school dental check, for example, and all those records, if they're available from school data sets, they can be very, very useful. Disease registries, in particular, say cancer registry, for example, has gained a lot of traction in these years. And in these cases, from the hospitals, from healthcare providers, disease data from individuals are collected, and they are stored in databases, and they're related with each other. So disease registries are also very, very important sources of data in identifying incidence rates and prevalence of specific diseases. The GloboCan, on the International Agency for the Research on Cancer, is a large data set which obtains data from cancer registries spread all over the world. And these are extremely useful things. Then there are health interview surveys that are conducted by ministries of health and health systems all over the world. And just to give you a sense of the different kinds of data sets that you can see in the context of New Zealand, take a look at this. You can see that there are a number of different data sets, well over 10, 15 data sets. You can see you can get data from The burden of disease studies, New Zealand Cancer Registry, hospital events, mortality collections, interview surveys, population statistics that are collected by the census data, surveys on disability. And see, a lot of them are on surveys. So we have got disability survey, New Zealand Health Survey, Maori Social Survey, and of course, census is also a very, very important unit of data. So just to give you a brief sense of how do people get data from individuals that are then pulled into the larger surveys and publicly available data sets. History is a very important source of health effects data, and history taking is an important activity on part of individual health practitioner or professional. Physical examination is also a very, very important part of record keeping, because physical examination will either refute or lead to differential diagnosis of a particular health condition. Physiological markers are the markers in which are measurements of specific molecules and concentrations of toxins that take place in the body as a result of being exposed to certain environmental agents. So physiological markers, for example, hemoglobin A1C or HbA1c as it is known as, is a very important physiological marker. It can also be a biomarker, by the way. A good physiological example of physiological marker might be forced expiratory volume in 1 second. This is usually done to measure whether a person has got asthma or not. And in this particular kind of a physiological marker, you can see that a person is asked to blow through a specific device, and the volume of air that is expired in the first second is measured. It gives you a signal of the respiratory capacity of an individual. Biomarkers, as we have mentioned, can be assessed in laboratories by first of all sampling blood or plasma or cerebrospinal fluid or urine, and chemical assays are conducted to find out, or chemical measurements are done in order to find out what are the different kinds of biological and biochemical agents that are present in those body fluids. And these are very, very important sources of data. In almost all circumstances, a specific diagnosis, etc., are arrived at by combining all these 4 sources of data, that is history, physical examination, physiological markers, and biomarkers. And therefore, these are important sources of information. Others include radiological markers, for example, X-ray that are taken, or ultrasonography conducted to get an ultrasonogram of the abdomen, for example, will yield important data about specific health states. So here, let's take a look at some simple things. For example, what happens, or what are the situations that you must understand when you start looking at or using questionnaires, for example. Questionnaires are quite commonly used both in case of surveys as well as in case of history taking by individual physicians and health providers. In general, any questionnaire that you use to get data from individuals should be very simple, and they should be unambiguous. In other words, if you are going to ask a question, you're going to only ask one question per item of interest. Do not combine 2 or more thoughts in one question. When you conduct surveys and when you conduct even history taking, avoid asking leading questions. So do not ask a question that can be answered by a yes or no answer, for example. That's a simple way of expressing how to avoid leading questions. Use words like how, when, etc., which will prompt your respondent to answer in more details. And any questionnaire that you draft that you either check or test or you deploy in the field must be tested for face, content, and construct validity. Face validity of a questionnaire refers to the readability of a questionnaire and the answerability of the questionnaires. Do your respondents, who are not experts, can they understand the questionnaire? Can they understand each of these items well enough to correctly answer those questionnaires? That is the kind of things that are tested in face validity. Content validity refers to the fact that if you provide your questionnaire to a content or domain expert, the content or domain expert should be able to understand or should be able to assess that all the necessary contents that are needed to be picked up to the particular research question that you are investigating are all there. If that is not satisfied, then there are issues around the content that you're putting across in this. Construct validity leads to the refers to the validity where the ideas or the theory or the hypothesis that you're testing using that particular questionnaire, they're all captured there. So constructs are like ideas or concepts that drive drafting of a questionnaire. Are all those constructs present? So that's known as construct validity. And once you have satisfied that your question passes good quality for face validation, content validation, and construct validation, then and then only can you deploy a questionnaire in a field and use that in the context of a survey. Lastly, let's come to take a look at what do we know about how environments impact organs and organ systems. Environments, our physical living environments, our social environment, our internal environment, our physical environment in particular, have got a lot of bearing on our well-being and on our health. Some of them are mediated by direct impact. For example, ultraviolet radiation impacts the skin directly. If someone drinks water which is contaminated with, say, E. coli, or food that is contaminated with E. coli, they come in direct contact with the gastrointestinal tract, and that is how environment directly impacts our health. High frequency vibrations and noise for prolonged period of time will damage our nervous systems. For example, people who use say mechanized lawnmowers, or say for example heavy machinery which vibrate a lot, say for example chainsaws, they expose their hands to heavy vibration, and that can damage the nerves. Heavy sound can impact and damage our eardrums. So all of these things are important, and they directly impact our. This is how these and some of the other mechanisms and are examples how environment directly impacts human health. Environment can have indirect impact on human health. For example, by altering the structure of our DNA, environments can impact us. And altering our metabolic states, environment can impact us. Sometimes, as a result of being exposed to some harsh environmental conditions, certain carcinogenic effects start rolling over, and that is known as environmental carcinogenesis. Environmental variables that can cause cancer, for example, altering the structure of the DNA, and then it sets the cell to start uncontrollably multiplying and infiltrating the tissues. Most of the times, however, most of our environmental conditions are non-cancerous. That is, cancers are rare, but other than that, there are certain environmental conditions that directly or indirectly can impact our health in many other ways. For example, if someone, there have been studies that have suggested that setting up of wind turbines can create a noise which is referred to as the whoosh, and that can annoy people and can lead to different kinds of mental and emotional health issues, and so on and so forth. And you can find several of these examples that can tell you that specific environmental conditions can impact our health indirectly as well. But these health conditions are not something that are cancerous. So this provides us with a quick summary or a brief tour of the role of health effects that can be explained by our environments. As we learned, health effects can be acute or chronic, and they can have different patterns of setting over time, and they can be minimal or they can result even in death for that matter. And health effects can be measured both at the level of the individuals or at the level of populations. And environments can directly impact our health and can cause cancer or non-cancerous effects, or and of course there is a genetic component to our health states, and environment and gene most of the times they impact together. There's hardly or rarely a case where only environment is responsible for our health state, or only genes are responsible for our health state. Usually it's always a combination of the two. So this was a quick introduction to health effects that you should know for environmental health. Thank you.